Hello, everyone. We talked about how the structure and the function of DNA was discovered in the previous lecture. What we're going to talk about from now on is the different processes that involve DNA. And so we'll talk first about how DNA is replicated, then how the information in DNA is translated first into mRNA, then proteins, and then we'll talk about a little bit of cell division, meiosis, mitosis, and some genetics as well. So let's start by talking how is DNA replicated and how ultimately it's organized, particularly in the eukaryotic cells. So this is the topics that we'll touch in this lecture. First of all, we talk about DNA replication. We'll talk about what originally were the potential models through which DNA could be replicating and how we narrow it down to what is called the semi-conservative model through the experiment of Mendelssohn, and Stahl. Then we'll talk about the process of DNA replications. So the role of this enzyme called DNA polymerase, the steps involved, and then we'll talk about the differences between DNA replication in prokaryotes and eukaryotes. And then finally, we talk about a few different topics. First of all, telomerase. This enzyme and the telomeres, those sequences at the end of eukaryotic chromosomes, which play a key role in ultimately controlling how long a cell should live for. Okay, so we'll talk about how telomerase work. We'll talk about how DNA is repaired, how the mistake introduced by DNA replication, and eventually external environmental factors are repaired. And finally, we'll talk about quickly how is DNA organized in eukaryotes. So let's start about how is DNA replicated, first of all. If you recall, Watson and Crick came out with this right-handed double helix model of DNA, which is based on two strands being complementary with each other. So as soon as Watson and Crick came out with this model, it was fairly clear to them and other scientists too that the nature, how the structure of the molecule itself could suggest how it could be replicated. You have two complementary strands. It's very fairly direct to imagine how if you separate these two strands, now each one of them could actually work as the template. So the strand that provides the information to make the other strand. Since you perfectly know that for any adenine, you have a thymine. So now if you have, or cytosine for guanine, if you have now a strand that has a T, you know that you have to put an A on the complementary one. So the nature of the structure of the DNA itself would suggest that DNA will be replicated basically by using one of these two strands as a template to make the next one. And then by doing that, you end up having two DNA molecules with each one of them with one of the old strands and one completely new strands. This is referred to as the semi-conservative model of replication. This is the correct one. That's indeed how it works. However, originally there were three possible models that had to be kept in consideration. Now, even if that model makes a lot of sense, in science you can't accept it just because it makes sense. You have to prove which one is correct. So in theory, there could be two other ways in which DNA could be replicated. So three in total. So the semi-conservative model, we already mentioned it. So you have that each strand is the template to make the new strands. And then so the two DNA molecule, as you can see them here on the top right, they will contain one old strand, the gray one, and one new one, which will be the right one. So both of them will be identical, but with half new and half old DNA. The second possibility is a conservative method, where replication happens the same way. So each strand works as the template for the new strand, but now the two new strands, instead of staying attached to the old strand, they get separated and they get put together. So now in this case, you would end up having one DNA molecule, which corresponds to the old one, conserve the old one, and then a completely new DNA molecule. So completely different overall. So it's very similar to the first one, but different in terms of endpoint. And this is referred to as the conservative replication. The other third model that you cannot exclude is what is called a dispersive model of replication or dispersive replication, where you have that in reality, for each molecule, you have the two strands, right? each one of them acts as a template, right? But then somehow the different new and the old one get mixed together so that you end up having that every single strand in the new molecule is a mixture of the new and the old one. So the new molecule is dispersed within the old one, and hence the name dispersive replication. You can see how these two steps, the conservative and dispersive particularly, will require extra step once the new DNA has been synthesized. They don't make sense, but again, theoretically, they could be all possible. How did we sort? So this is just a slide that shows you basically what I just said, how the conservative replication would work. One DNA molecule becomes the strand, the template strand for the other molecule, and then the new and the old one will stick together. So each DNA molecule has half of the old, half the new DNA. Differently from the conservative replication, where you have that in the end you end up with a DNA molecule which is completely new and one which is completely corresponding to the old one, and dispersive replication where you have that both DNA molecule would end up being a mixture of the new and the old DNA. And these obviously will carry on to the next generation. We'll see in the next slide when we talk about the mesoscale and stall experiment, how it's important also how it will go in the next generations. So let's talk about how finally we had an evidence supporting the semi-conservative model of replication. This was done by these two scientists, Mendelssohn and Stahl, which basically did a very clever, again, experiment where they used bacteria of the species E. coli, Escherichia coli. And what they did, they found out, they first developed a method to recognize new and old DNA. How did they do that? Well, DNA, as you know, is a nitrogen containing molecule, right? So what it means is that now if you take DNA and you put it with any isotope of nitrogen, and there are different isotopes out there. The main one is actually N14, and there is N15 as well. They would include this nitrogen. So if you take some bacteria, here you have some bacteria growing in this flask, and you replace the nitrogen there with nitrogen 15. So you have only this isotope of nitrogen. This has an additional neutron compared to nitrogen 14. What happens there is that this bacteria would take nitrogen 15 as any nitrogen and would include it into the DNA. So if you let it go for several generations, you would end up having bacteria now which have DNA, so that the nitrogenous bases in their DNA will all contain nitrogen 15. Great starting point. So now you have DNA with nitrogen 15. It's non-radioactive. Okay, just to make it, they're not working with radioactivity, they work in a different way. Great. So now you know how the original parental DNA contains nitrogen 15. And now, to see what happens during replication, what you do, you let this bacteria, N15 bacteria, replicate for 1 or 2 generations in the presence of a different isotope of nitrogen, nitrogen 14. So at this point, when replication is happening, instead of entering nitrogen 15, the newly synthesized DNA would have nitrogen 14, right? So you get that going for 1 or 2 generations, and then you see what happens. How can you see how much nitrogen 14 or nitrogen 15 you have in the new molecules, which will point to how replication happens? How can you do that? Well, you can do that by taking advantage of the fact that nitrogen 15, having an additional neutron, is slightly heavier than nitrogen 14. So now multiply by the thousands of nitrogenous base you have in the DNA, you can see how overall a DNA containing nitrogen 15 will be heavier than a DNA containing nitrogen 14. How can you see that? Well, you can see that by extracting the DNA from the bacteria and basically letting it precipitate. You can do that leaving it on a bench. It would take forever, years and years to see that. But you can speed up that process by doing what we call centrifugation. Centrifugation is a process where you put any sample, in this case will be a DNA extract from the bacterium. You put it inside a test tube and you increase gravity by spinning this sample with a certain amount of rotation per minute. So just to give you an idea, with DNA, usually you work with 10,000 to 13,000 rotation per minute. So this thing will do 10 to 13,000 rotation every minute. By doing that, you're increasing the gravity that this DNA inside this test tube is undergoes to by thousands, okay, depending on the radius of the router too. So it changes, but you're talking about accelerating gravity by thousands of times. So that process that would take years, now it will take a few minutes. So how does this translate? So if you take the DNA now from this bacterium with DNA with nitrogen 15, other bacteria with DNA from nitrogen 14, or a hybrid, now if you take their DNA and you spin it down in a particular medium, it's not in water, okay? You need cesium chloride and so on, something that allows to distinguish the density and the weight of small molecules like them. You will get that you can actually visualize the weight of the DNA. So if the DNA contains all N15, like in this case here, it will be much heavier, so it will precipitate faster, so it will be closer to the bottom of your test tube here, than if it contains all N14. And now if you have a hybrid molecule with some N15 and N14, it will be in the middle. Great, so now mesenchymal and Stahl, they have a way to follow what's happened to DNA. How is DNA replicated will be visualized by looking at how heavy it gets after 1 or 2 divisions. So let's go through their experiment. So these are the possible models. Okay, now we'll go through the results that they got. So they start with this bacteria. They all contain N15 DNA. And they let that go through one cycle of replication. E. coli replicates in around 20 minutes. So it's a great system, fast system. Then they extracted DNA. So what would you expect to get if DNA replicated as a semi-conservative molecule? You would have that the two DNA molecules, as we discussed before, would be a mix of the old and the new one, right? So since in this case, this first replication has been done in N14, now you should have that the weight of each molecule should be half the weight between a molecule that has all N15 and a molecule that has all N14. So it should be basically giving you a band like this one or in the middle right here as a reference. If it was conservative, instead, I hope you can see it, so you would have one DNA molecule that will be all completely containing N15, the original one, the parental one, and one completely new. So what you would expect to get is two bands at the extreme opposite. One band, very light, the N14 that the new DNA would correspond to, this one right here, and the old DNA, very heavy, N15, N15, two bands. So already you would expect a result different from semi-conservative replication. What would happen if you have dispersive replication? You should get two DNA molecules that are a mix of both. So overall, these two molecules should have most likely a similar weight, right? And therefore should give you a band, should have a weight which is intermediate, similar to the one you have with semi-conservative replication, right? So this is what they observed. What Messer and Starr observes that indeed they were getting, after one cycle of replication in M14, they were getting only one band, which was, in terms of weight, intermediate between the pure N14 and the pure N15 DNA. Therefore, supporting potentially semi-conservative replication, but also dispersive replication. So now, how can you distinguish between these two models? Right now, this evidence is not enough to support the semi-conservative replication or the other one model. How can you do that? Well, by letting the bacteria go for a second round of replication. So what would happen if you have a second round of replication still in N14? Now, let's look at the semi-conservative replication. What you would get is that each of these molecules act as a template, again, for the new DNA, and then one of the DNA would have a mix of the old, which is actually still coming from the original DNA, and newly synthesized strand. The other one now will be this new strand in the first replication will be also the old strand in the second generation. So you will get, at this point, four different ovally DNA molecules in this present. So two of them with a weight that should be intermediate, and 15 and 14, and another one that now should be completely containing N14 DNA. So you would expect to get two bands, an intermediate one, and a very light band. You should not get N15 anymore. Now, if you look instead of the dispersive replication, now, since the new molecules are a mix of the old and the new one, you should still get probably one band or a smear of different bands at this point with a mixture of N14 and N15. So you should not get two clear bands, but you should get something in between here, most likely a little bit higher because at this point you should have a little bit more N14 incubated. But you should not get two clear bands, N15, 14 and N14, N14 bands. You should get something, one single band in between here. Well, what did they observe? Indeed, they observed the two bands that you would expect with a semi-conservative replication. So supporting that DNA indeed replicates by semi-conservative replication, since you didn't get a smear here in the middle, which is what you would have expected with the dispersive replication. So now we know, we have evidence done by Meselson and Stahl that DNA replicate in a semi-conservative replication. Let's go through the steps. How does DNA actually replicate? I want to stress one thing. What we'll talk about mostly here when we talk about the different step is how it works in prokaryotes. It's the simplest model, but this applies overall also to eukaryotes. And we'll talk about that later on, about the differences as well between prokaryotes and eukaryotes. First of all, let's talk about the enzyme that do the replication. The enzymes that are important to synthesize the new DNA strands are called DNA polymerases. As the name says, what they do, they polymerize, so they add together different nucleotides, in this case deoxynucleotides, because we're talking about DNA. All deoxynucleotides can be represented as DNTPs, where N is a variable, it could be adenine, thymine, and so on. And S stands for plural, so D for deoxynucleotides. So what DNA polymerase does, it polymerizes these deoxyribonucleotides and into DNA, adds them together, forming the new strand. Now, the main ones in prokaryotes are DNA polymerase 3 and polymerase 1. There are more than two present in eukaryotic cells. Now, how does it work? So how does the polymerase know how to add which nucleotide add is by reading the original strand of DNA that works as a template. So it's looking between brackets, okay? So it's sensing what it is on the template strand and adding the complementary nucleotide for that specific base in the template strand. And then it does that by adding the nucleotide to the 3' hydroxyl group of the growing strand. Do you remember? So as I mentioned, when we're talking about the orientation of the strand, when we're talking about DNA structure, I was telling you the direction, the 5' to 3', 3', 5'. By adding the next nucleotide to the 3', it gets the new strand of DNA to grow in the 5' to 3' orientation. You can see it better in this picture. So this is the template strand. This is the old DNA that gets copied into the new one. The old strand copied into the new one. So how does DNA polymerase work is by adding one nucleotide at a time to the 3' hydroxyl group of the growing DNA. And so it does that by taking a deoxyribonucleotide 3-phosphate. So you get the energy as well here. These two phosphate groups are removed. There is energy. This phosphate group is transferred with its energy to the hydroxy group on the carbon 3' of the deoxyribose here. And then this will become the next nucleotide to attach the next deoxyribonucleotide to. So the DNA at this point is growing in this direction, 5' to 3 prime. That's the orientation. Second thing I want to point to you is how the template strand dictates what nucleotides will be added. So now there is a timing here based on the Chargas rule and the matching between adenine and thymine. You know that the next one is going to be adenine. DNA polymers can sense that in terms of stability of bond, of hydrogen bonds. And then here there is a guanosine, so it would add a cytosine here, and so moving on. So the template gives the information to the DNA polymers to add the next nucleotide to the growing strand in the direction of 5' to 3' by attaching the next nucleotide to the hydroxy group in the 3' of the deoxyribose. So this is how the DNA polymerase will look like. So DNA polymerase looks like basically a hand. Imagine like so you can take your hand, looks like this. So, and basically what it does, there is another protein, we'll see, sliding clamp, that helps polymerase stay attached to the DNA, but basically it has an active site that is formed by this folding here, which is where the newly synthesized strand will be in with the old one as well. And that's where the new nucleotide is also brought in. And so that's where the new nucleotide, the triphosphate form of the new nucleotide, and the growing strand are brought in. That's the active site, and that's where the reaction happens right there. Now, the DNA polymerase doesn't work on its own. And again, what we're talking mostly about here is DNA replication in prokaryotic cells. There are differences with eukaryotes, but DNA polymers in general do not work on their own, but they are kept attached to the DNA by these other proteins, which is called the sliding DNA clamp here, which actually forms like basically a circle that goes around the DNA, keeping this DNA polymers attached to it. Now, notice, just pointing to use or practicing something that we learn when we talk about proteins, how this protein, for example, contains alpha helixes here and beta plated sheets here, represented by these ribbons here. Now, let's go through the step in DNA replication. So conveniently, DNA replication, we'll see also translation and transcription and so on, they are divided into steps. So when it comes to DNA replication, you have 3 different steps that you can recognize. The first one is called the unwinding of the DNA. DNA polymerase to work needs to have a single strand DNA to act as a template. So the DNA has to be open. Okay, and that's what happens, as I say, during the unwinding of DNA. Once that has happened, now DNA polymers can act and form the new DNA. That's referred to as the elongation step of replication. And then at the end, you'll see this will lead to the formation of RNA-DNA hybrids first. With the RNA, that has to be replaced with DNA. Everything gets joined together, and that's referred to as the termination step of replication. Let's start with the unwinding. So what happens in the first step during unwind? So the DNA gets open. This doesn't open on its own and everywhere in the DNA. There are regions. In prokaryotes, there is only one of these regions. In eukaryotes, there are multiple ones because the size of the chromosome is much larger in eukaryotes. But there are regions called origin of replications. These are regions that are fairly rich in adenine and thymine. Not surprisingly, adenine and thymine, they join together with only 2 hydrogen bonds. So by having a lot of them in these regions, you make these 2 strands easier to separate. So this region called origin of replication, that's where DNA replication starts. This opening doesn't happen on its own, but is mediated by this enzyme called DNA helicases, which basically bind the 2 strands and apply the energy necessary to open it up. Once the 2 strands are opened up like this, forget about this part. This is already into elongation, this part here, but you would have the 2 strands that get separated. And then to keep separated, these 2 strands, once the helicase moves on, they would tend to join together again. So there are actually proteins called single-stranded binding proteins here that do help to keep these two strands separate. They bind to each individual strand and prevent them from forming the hydrogen bonds again. Not only that, now there is another protein, another enzyme that's important for this step, which is one that prevents basically the DNA to break far away ahead from where the opening is happening. So now, DNA is opening. This establishes two replication forks. Each of these half here will be referred to as a replication fork. You see, when we talk about DNA replication, so the elongation, we'll be referring only to one of these forks because it's the same thing that happens on the other one in the end. But so establish two replication forks here. But now, while the DNA is opening here, is actually applying a lot of force on the DNA that is still double-stranded. Just to make you understand that, imagine you take two strings, okay? Join them, make a knot at one end and leave the other end open. Form a nice helix there. And now you have one end that is tied, the other one that is open. You take the two strands and you try to open them up. Pull, pull, pull. At a certain point, you see that you are creating a force towards the end that is joined. So imagine this being your string, the end that is joined. Here's where you are pulling up, up, and up, and up. You're creating a force. These two strands will get super coiled here. And so this creates a strand that will cause the DNA actually to break. How is this prevented? It's prevented by these enzymes called topoisomerases. And what they do, they just cut one of the two strands and let it uncoil around the other one. Same thing, if you take your two strings, cut it towards where they're joined, you will release that tension. Once that has been released, you can cut it, you can join it again, as the topoisomerases do, and now you have less force applied to the double strand. That's the role of topoisomerase. They prevent this excess tension that will be building up with the DNA opening. So three key components here. Helicase help the DNA opening at the origin of replication. DNA binding proteins, single-strand DNA binding proteins, they do prevent the DNA from rejoining, and topoisomerase prevent the DNA from getting an excessive tension and breaking apart in the region that are not open yet. So once it is done, now you have established two replication forks, one and one. We'll focus only on one of them now in the next process, but what we'll discuss for one replication fork applies also in the other one, to the other one as well. The next step is what we call elongation. Elongation is that step in which the DNA gets, actually the old strand gets copied into the new one. Now, there is a problem with that, is that at the beginning, once the origin of replication has opened, you have nothing. So you have the two strands separated, but there is nothing as part of the new strand. Why is that a problem? The problem is that DNA polymerase can only do one thing, which is to attach a new nucleotide to an existing nucleotide. So if you are in a situation like this, where there is no nucleotide here, where you can attach a nucleotide, nothing would happen. You can take DNA, open it up, and you add DNA polymerase, nothing would happen, even if there are the nucleotides there. It cannot polymerize because it needs something to attach the first nucleotide to. So how is this prevented is by the action of another enzyme. In this case, this is an RNA polymerase. RNA polymerase, as the name says, polymerizes ribonucleotides, not deoxyribonucleotides. So it forms RNA instead of DNA. So this RNA polymerase, called RNA primase, doesn't need a starting point. It can actually start adding ribonucleotides based on a template sequence on its own. And so this enzyme, what it does, it creates some short primers, up to 20 nucleotide long. It's longer than what you see here, starting from nowhere. So it takes some ribonucleotides and joins them together, complementary to the DNA strand here, creating this RNA primer, which then DNA polymerase, in this case will be DNA polymerase 3, can start working from to copy the rest of the DNA, which is what you see here. So these will be the RNA primers formed by the RNA primase. Again, doesn't need anything to start from, so it can do it anywhere. And then DNA polymerase has finally an hydroxyl group here to start attaching the first deoxyribonucleotide, then the second, and so on. So it will elongate this RNA primer. As a result, you come out, as you can see, with RNA DNA hybrid molecules. We'll see how in termination this RNA is replaced. So DNA polymerase now can act and move on. Now, depending on which strand for each replication fork, so this is one replication fork, depending on which strand you are looking at, you can see how one of the two templates can actually be continuously, starting from one RNA primer. Look at the one on the top in this case. So the RNA template here is in the 3' to 5' orientation. So now you have RNA primase has done a primer, produce a primer, RNA primer here. And now DNA polymerase can start from here to copy the DNA. Now this DNA will keep opening up. You can see how it can keep going. Doesn't need any other RNA primer to be formed because we'll always have a 3' hydroxy group at this point to keep going. So this strand can be copied continuously and is referred to as the leading strand. In the opposite strand instead, so you have now these RNA primers that get extended by DNA polymerase 3. Now you have this, but then when the DNA opens up here, you can see that now DNA polymerase cannot continue because it cannot work on the 3' to 5' orientation. It can only work in the 5' to 3' orientation, right? And so what it means is that while this keep opening up the replication fork, you need more primers to be formed. So this strand here cannot be copied continuously, but can be copied discontinuously, producing a series of fragments called Okazaki fragments, which have DNA and RNA formed here. So one strand, the leading strand, gets copied continuously because it can keep going in the 5' to 3' direction. The other one instead will need more and more primer be formed, and this is referred to as the lagging strand of DNA. Okay, and this will keep going. So this elongation step, where you have the RNA primers forming some primer, DNA polymerase 3 extending these primers, will keep going with the replication fork opening up more and more and more until all the two strands have been separate. These original strands have been separate. Now, the problem you have is that once this replication fork, the same thing would happen to the other replication fork. I hope, though, you can imagine how the leading strand in the other replication fork would actually be the lagging strand, and vice versa. The lagging strand in the other replication fork will be the leading strand. So if this keeps opening up until the two original template strands have been opened up, you have copied all the DNA, but you have a problem. Is that the DNA has been copied, but you still have RNA in that DNA. That would not work. You can't pass it to the next generation. You need to replace these RNA molecules, and this is what is done in the last phase, the termination. How is this done? This is done by a different type of polymerase called DNA polymerase 1. It's different from DNA polymerase 3. DNA polymerase 3 is the one that extends these RNA primers, keeps going. DNA polymerase 1 instead, unlike DNA polymerase 3, has an extensive 3' exonuclease activity, which basically allows it to go in 3' to 5'. We're talking about the opposite direction now than the polymerase activity. And when it goes in 3' to 5', so it goes in reverse. Imagine you take your car, you put it in reverse. It's going backward. It can actually remove the ribonucleotides in the RNA. So it will bind to these RNA primers here in red, here. And then in the 3' to 5', so this is the 3', this will be the 5'. In this direction, it can actually remove the primers, going in reverse. Once it has removed all the primer, now it can go back, to the 5' DNA polymerase activity. So with the exonuclease activity 3' to 5' removes the ribonucleotides and then goes back 5' to 3' with the DNA polymerase activity, replacing basically the ribonucleotides with deoxyribonucleotides. So replacing this RNA primer ultimately with a DNA molecule. The problem you have is that once it reaches this point here, so it has replaced, imagine this red sequence, this RNA being replaced all with DNA, so it will be in blue by the end. Now it cannot do the last joining here because it would have to add these last nucleotide to a 5'. Here you would have a 5', a phosphate group. So it cannot join to a 5'. Remember, DNA polymers can only attach the nucleotide to the 3', not the 5'. So how is this gap filled? So this last phosphodiester bond is done by another enzyme called ligase. Ligase, what it does, it forms this last phosphodiester bond necessary here to finally have a continuous DNA molecule here. Once this is done, DNA replication has been terminated. You would end up having 2 strands here, all made by deoxyribonucleotides. So 2 full DNA molecules that can then be divided into 2 different cells in the cell cycle. So summarizing the process again, there is a lot of steps here. Let's summarize first the process itself and then all the enzymes involved. Summarizing the process, first step is unwinding. The DNA gets opened up at the origin of replication through the help of these enzymes called helicases or DNA helicases. They separate the 2 strands. Now, second set of protein, the single-stranded DNA binding proteins, or as BP here, BPS will be, they keep the separate strands, the 2 strands separated, and then topoisomerase here release the tension that is building ahead of the replication fork. After that, elongation takes place. But before DNA polymerase 3 start forming the new DNA, you need the formation of some RNA primers, here in red, by these RNA polymers called primase. Primase does these RNA primers, and then DNA polymerase 3 can use the hydroxy group at the 3' of this primer to start copying the remaining part of the DNA. You have a leading and a lagging strand. The leading strand is going to be copied continuously as the DNA keeps opening up. This will be topoisomerase here helping, releasing the tension. This can go continuously, is the leading strand. The other one will need more and more and more primers to be formed as the DNA opens up, is the lagging strand. This process will keep going until all the DNA gets copied, but then you still have RNA molecules in there. Keep in mind that all of this is happening at the same time, these phases. I hope you can see this, how it's separated into different steps, but these things are not really divided so clearly. But what happens in the last step, so the RNA that is still left behind, so these blue molecules in this picture here, sorry, these, the RNA molecules, okay, would have to be replaced with DNA. And this is done by different type of polymerase in the termination phase, which is DNA polymerase 1 right here, which basically replaces the RNA molecules with DNA, leaving a little gap between the last nucleotide it adds and the one that was there, which is sealed by DNA ligase, which forms the last phosphodiester bond. After that, you end up having a final DNA molecule completely copied into another one. So these are all the enzymes we talked about. Helicase, that opens up the DNA. Single-stranded binding proteins, they keep the DNA open. Topoisomerase, and there is primase. DNA polymerase 3, different from DNA polymerase 1. DNA polymerase 1 is the one also that removes the RNA, replaces with DNA. The sliding clamp that helps DNA polymerase 3 and 1 to stay attached to the DNA as well. And then you have DNA ligase that seals, forms the last phosphodiester bond. So let's add a few details here to this process. So you know all the key steps now. There are some differences between eukaryotes and prokaryotes. In general, the process itself tends to be faster in E. coli compared to eukaryotes. Eukaryotes have a more complex type of DNA with histones and so on, which slows down the process. So in prokaryotes, these process goes at a rate of about 500 to 1,000 nucleotides per second. It's fairly fast. Whereas in eukaryotes, it goes for about 50 to 100 bases per second. So it takes a longer time overall. You can see how also these could represent, if you know this process very well, it could be a great target also for the design of drugs. Several antiviral drugs are indeed made to target this process here. So, and they're made by using nucleotides analogs like the one you see here. So you have different types, sidosylfovir and so on. There are many, many types. As you can see, what they do, they simulate a nucleotide. So they would have a nitrogenous base here, a phosphate group, but they're missing the deoxyribose here. So, but because of this, they have an hydroxy group here, which simulates the 3' hydroxy group. They have a phosphate group here. So if you introduce them as a triphosphate ones, they actually can be included in the new strand while it's synthesized. But by missing the remaining, so by missing the deoxyribose here, then they would stop the continuation of the process of synthesis of new DNA. So they get integrated in the new strands, but then they stop the growth of that strand because there is no deoxyribose that the next nucleotide could be attached to. There other difference between, other than the speed between eukaryotes and prokaryotes, is in the way the process is done. The steps are similar. The DNA polymerase is slightly different, but we won't get into that. But one thing you can imagine is how inefficient it will be for eukaryotes if this process rely only on one origin of replication. The reason being that the DNA in eukaryotes is thousands of times larger than the one in prokaryotes. You're talking about billions of base pairs compared to millions. So 1,000 times larger. So if you go on only one original replication, it would take forever. The other thing is that also the DNA is divided into multiple chromosomes. So you will need at least one for each chromosome. But in reality, you have more than one for each chromosome too because of their length. So there are multiple original replications. Each one of them starts on its own. That's exactly the same process that we discussed. And now you would have the replication fork from one original replication will keep expanding one from another one, and they keep expanding opposite to each other until they join together, basically, right? In a circular DNA instead, imagine this being a circular DNA in a prokaryote, this will keep going until this fork and this fork starting from one original replication, again in prokaryotes, these two replication forks will meet at the opposite end of this circular DNA molecule. In eukaryotes, as I said before, they have large genomes, multiple chromosomes, so they need different origin of replications. And then they keep going until they get to the end of the chromosome or meet each other. This process you see when we talk about the cell cycle. The cell cycle is that cycle of events, like replication of organelles, of ribosome, replication of DNA, that basically leads one cell to divide into two. Obviously, a cell to divide has to go through different steps. There is a step where it will grow, it will multiply its organelles, like its mitochondria, its whatever, its endoplasmic reticulum, ribosome, and so on. Then there is a phase called the S phase. That's where actually the DNA gets replicated using the process that we discussed before. And after that, this DNA will get divided in the process called mitosis or meiosis, depending on the cell we're talking about. So what we discuss here, keep this in mind. We'll talk about the cell cycle in one of the next lectures. This is happening in the S phase. Okay. As we said before, now in eukaryotes, you will have multiple original replication. These will lead to different replication forks moving in the direction of each other. Some of them move towards the end of the chromosomes. and therefore leading to the end, to the full duplication, the fusion of the replication forks and the full duplication of the chromosomes. I'm going to stop here for this part of the lecture. Then the second part, we'll talk about telomerase, DNA repair, and the organization in eukaryotes.